Hello, my name is Duane Johnson. It's July 23rd, 2018. This is my lab assignment on bacteria identification through functional media motility testing. So let's just get to it. We're going to start with the motility test agar. We're going to start with the E. coli. It's labeled E. coli. Start by taking off the cap again. Then we're going to pass it through the flame a few times. And then we're going to get to our E. coli plate now with all the colonies that are still in it. And I have two actually sterilized autoclaved probes that I'm going to be using instead of the paper clips. I also flamed them until they were red hot and then started soaking them in the alcohol. So they've been soaking for a while. Get the one. So these should be very, very clean. Just gonna wait till this is a bit dry, more dry, completely dry. In that step, I'm gonna get the E. coli ready. So it's the E. coli. I'm going to be getting a few of the colonies. I'm gonna try to get some of the colonies with the probe, and then I'm just gonna dunk them from the probe into the motility testing agar. And then we're going to incubate them for 48 hours afterwards. So this looks very dry, so we can begin. I have very large colonies, so I hope this my technique is proper for this. Just gonna, oh yeah, get some of those colonies there. Oh, that's cool. Maybe over here too. Now, it says to dunk them in and remove it in the same pathway as it was dunked in. So I'm going to try to do my best. Small opening, but I'll do my best. Is this all breaking off? Cool. And I'm just going to toss this straight into the undiluted bleach, because that's gross. Now, go ahead and put the cap back on that and reflame this a few times, couple passes. I'm gonna put this cap back on. Wow, look at how they just float. That is super cool. They just float there. So we have one more to do. This is my little makeshift test tube rack. Next one. Motility test agar with the S. epidermidis. Same process, same procedure. Have the probe. Wait for the probe to dry a bit. Still see some liquid in there. I have quite a few colonies in there too. My Staphylococcus epidermidis. I think my culture plates were probably my plates are probably the best so far. Out of all these experiments, I really like the plates. So, yeah, we're dry. So I'm going to gather a few colonies and dunk them down into there. right there in the center. That's awesome. That looks really cool. Wish I could do that with other stuff to look at it. Now we will incubate those motility test agar tubes. I'm gonna be putting my culture plates back in the fridge, and that is part 1 of my bacteria identification through functional media motility testing.